Okay, guys, welcome back. And today we're going to be finishing up the blood and starting the heart lecture. But this is all part of the cardiovascular system that is itself part of the circulatory system. So this very important slide that we have here. So you should be able to look at this figure and interpret and describe what's happening. So we begin from left heading to right. So the most immature cell is to the far left, and that is the pluripotent stem cell. From the name, you can tell it's a stem cell because it stems and grows and develops and differentiates. Remember, we were talking about, Pokemons, they evolve, but we don't say a monocyte evolves into a macrophage. That doesn't make sense. We say it differentiates. So the pluripotent from the name, potent is potential. It has multiple potentials. Can you see the different potentials that this cell has? The first potential it has, it can become a eosinophil. The second potential this very same cell has is that it can become a basophil. The third potential, it can become a neutrophil. Fourth potential, a monocyte that can later on differentiate into becoming what? The monocyte. The monocyte has the potential of eventually evolving and becoming and differentiating into a macrophage. And you have these lymphocytes, B lymphocyte, T lymphocyte, and natural killer cells. So what I'm trying to say here is that those are all different types of really equipped. These cells are ready to fight. All of these mature cells, they are ready to fight bacteria, microbes, viruses, parasites, different types of fungi. So that's like the military, the Navy, the Marines, they are ready. They have been trained. But they all came, it's like a person, a human being who is immature, goes to one camp, ending up becoming a Navy SEAL, going to another camp, becoming a Air Force pilot. But it's the same person. That's the same thing. You have a one stem cell that can become any of these different mature cells. Which one of these cells is going to be specialized in combat with bacteria? The neutrophil. So that's why when you do blood work and you see so many neutrophils in your blood, you know you have a bacterial infection. That's how we tell there's a bacterial infection if there's so much neutrophil. So what causes this particular cell to become one of these different mature cells? If you see the arrow, there are different pathways, different camps, different training camps, and then they're going to graduate and become all of these different specialized mature cells. What does the arrow represent? We discussed that yesterday. Remember the concept of the cytokines. So cytokines are molecules, are proteins that once they bind to a receptor on the surface of the cell, they're gonna push the cell in one direction. So if your body wants eosinophil, it's going to secrete eosinophil cytokines, and that cytokine is going to make the pluripotent stem cell differentiate into a eosinophil. It's gonna make the basophils, it's gonna make this pluripotent stem cell, pluripotent, plural, multipotent, multiple potentials. A potential to become eosinophil, potential to become basophil, potential to become a neutrophil, potential to become a monocyte, and the potential to become either B or a T lymphocyte or a natural killer cell based on the correct cytokine. So if you take this pluripotent stem cell and you give it a basophil cytokine in lab, it's going to differentiate and it will definitely become a basophil. So it is the cytokine is the one who is responsible for guiding the growth of this pluripotent to guide the pluripotent stem cell into becoming one of these mature cells. Is that point clear? That point is very, very important. Okay, so from the name, leucopoiesis, poiesis, leuco. So from the name, what's a leuco? Leukocyte. Yeah, so the leukocyte is the white blood cell. Poiesis, what's poiesis? You know, you can hear the term hematopoiesis. So poiesis is the genesis and the making. So leucopoiesis is the making of leukocytes. Hematopoiesis, what is that then? So from the name, leucopoiesis, poiesis is the genesis and the making. Leuco is the white blood cell. That's the making of white blood cell. What about hematopoiesis? What did that mean then? Yeah, the making, the genesis of red blood cells. Okay, so now let us go back into our white blood cells. We discussed that our blood has different types of proteins, and they are the albumins, globulins, and fibrinogens. What did we say about nogen? If something ends with nogen, like angiotensinogen or fibrinogen or plasminogen, we will see that today, plasminogen or trypsinogen. What does that represent? Nogen, if it ends with ogen. So clotting factor if it has a fiber in front of it, so fibrinogen. But the concept, if you have something that ends with ogen, it means that it's inactive, it's off, it's inactive. So if you want to activate fibrin, so fibrin is the active form. That's the one that's gonna make the blood clot. Fibrinogen will not. So you need a protein that's gonna cleave and break off this ogen suffix, and you end up with precursor of fibrin threads. They never said fibrinogen threads because they have been now activated. So you cleave and you remove the ogen. You will see this again. We will talk about this concept again. Whenever you see ogen, it means that we are, activating the molecule. So we also discussed this concept of proteinemia, hypoproteinemia. So can someone explain how could this child with Kwashiorkor's disease develop this enlargement of the abdomen, this swollen abdomen? How did that happen? Yeah.
Water from the abdomen and out of the limbs.
Out of the limbs?
Or it's like it's leaving the arm, going into the.
It's leaving the blood. Remember, the scale has to be balanced. Okay? It has to be equal. The scale has to be equal between the blood and the tissue compartment. In this case, is the abdomen tissue. So the water here must equal the water in the abdomen. And the osmolarity of the blood must equal the osmolarity of the abdomen. What happens is because they are so impoverished, there is no protein. So now it's supposed to, the protein in the blood must equal the protein in the tissue. So what happens is because of the poverty, the protein in the blood is so low compared to the protein in the tissue. So now it's no longer balanced. There is more protein in the tissue and less protein in the blood. So now water is going to go from where to where? The water is going to go from the blood, because there's less protein, into the tissue of the abdomen, because water moves from an area of a lower osmolarity to an area of a higher osmolarity. Remember when we drew the 25 milliosmoles, 250 milliosmoles, 2500 milliosmoles, water is going to go from 25 to 250, but if it encounters 2500 milliosmoles, it's going to go to that one. So water is always going with there's a lot of osmolarity. So that's what leads to the abdomen swelling. Okay. So this red blood cell, also known as the erythrocyte, and many of us learned about the red blood cell even in living environment from high school. So you know that the red blood cell is carrying oxygen, but it is also carrying carbon dioxide. Can the red blood cell carry both at the same time, oxygen and carbon dioxide? That's not going to happen because you cannot be going in an uptown subway trip at the same time somebody's trying to go downtown, but they are on the same track. It does not make sense. So you have two trips, we said. One trip is uptown going to the lung, and you have the tissue compartment. So the tissue is utilizing oxygen all the time and burning oxygen to get the waste carbon dioxide. So carbon dioxide is taken up to the lung and then it gets expelled out, and then oxygen enters the lung and it gets sent down to the tissue. So both trips bringing down oxygen or sending back carbon dioxide, this is all achieved by the red blood cell. You know, this is anatomy and physiology, right? So we are always concerned with structure of things and function of things. So you must understand that the red blood cell is biconcave. What does biconcave mean? Biconcave means that it is depressed in both directions. Bi, two, one, two. Biconcave. It is depressed in both directions. Why is it depressed in both directions? Because that is the most favorable structure to allow this red blood cells to maneuver inside the blood vessels and they don't get trapped. In fact, in the spleen is known as the spleen is known as the graveyard of your red blood cells, the RBCs. Why? Because the spleen has sinusoids. What are these sinusoids? Sinusoids are small spaces that are like a challenge. They present a challenge for these are fluid filled sacs or spaces, and they present a challenge for the red blood cell. So the younger red blood cells are able to maneuver through the sinusoids so easily. But the old worn out, the old damaged, defected, worn out red blood cells, they get trapped. And once they get trapped, there are macrophages in the spleen that destroy them and get rid of them. We will see that the red blood cell has a life cycle. People have a life cycle. You're born, you're an infant, then you're a toddler, then you're a teenager, then you're an adult, then you're very old, and then you die. Same thing, red blood cell. So the red blood cell is gonna at some point in time is gonna die. Where is it gonna die? It's gonna die in the spleen, in the sinusoid. How? When it fails to maneuver properly. Why? Because it lost the biconcave structure. So it's not excellently biconcave like that. It loses it. So now it becomes, it loses the ability to maneuver inside these red blood cells. Another problem is sickle cell anemia. So why is sickle cell anemia a problem? Sickle cell anemia, the red blood cell does not look biconcave. It looks like a sickle. You know that structure? This scary thing that they always have in Halloween and is holding this thing like that. You know what I mean? This? You know this? Yeah, that's where they got the sickle from. He's supposed to be cutting the grass with that thing. Yeah, so that's what they named it after. Because it really actually does look like in the microscope, it looks like this sickle. This actually is so sharp, it even injures the blood vessel that the blood is going through. And it tends to clump up a lot of red blood cells, and that makes a red blood clot. So this structure here, this sickle structure, is really dangerous. It's not supposed to look like a sickle. It's supposed to look like what? The red blood cell.
The monoconcave.
Yeah, it's supposed to be looking like biconcave structure. So what's this sickle cell anemia? Well, what is anemia? Anemia is when there is a drop in the count of red blood cell. When you do not have enough red blood cells. So now there's hypo. So we don't say hypo red blood cell. We just say anemia. There are different kinds of anemias, but one of them is a sickle cell anemia. Because even if you have the proper right number of red blood cells, but they are not biconcave, you really don't have an actual red blood cell. It's a damaged, defected red blood cell. Okay, why do they sickle? So if a person who has sickle cell anemia, if you take a random blood sample from the person with a sickle cell anemia, will they look like that in the microscope? Let's assume that I, God forbid, have sickle cell anemia, and they take a blood sample from me right now, and they look under the microscope. Is it gonna look like that? It's not. It's gonna look biconcave. But if they ask me to get on a treadmill and start running, then while I'm running, they take another blood sample. Then they look at it in the microscope. Is it still gonna look biconcave? No, it's gonna sickle now. Why? They sickle when it's too hot, or maybe when the weather is so hot, the blood is gonna sickle, or when there is an oxygen demand. So your red blood cells, when you need them the most, they're gonna start sickling. If you have sickle cell anemia. So patients who are suffering from sickle cell anemia, they have this problem not all the time, only when they need their red blood cells the most. That's when their red blood cells start to sickle. Is that point clear? Okay. Okay, that's a very important structure. That's the hemoglobin. You see, hemoglobin has four subunits. It's a globular protein. It's a globular protein that is a quaternary structure. You can see the four structures. Two twin. You have two set of twins. The first twins are two alpha, and the other twin are two beta. So two alpha globules and two beta globular structures. Each globular structure, you see this red heme group, that is where your oxygen binds. So you must understand that hemoglobin has four proteinaceous structures. It's a quaternary structure made up of two sets of twins, two alpha structured and two beta, and each one, whether alpha or beta, each one has that red heme group to which oxygen is going to bind. The heme group has an iron in the ferrous state, that is an oxidation state of +2. You see, that's a ferrous ion, and that's the one that's going to bind to the oxygen. So the globins, we said these are four protein chains. Two of them are alpha and two are beta chains. Okay. So we mentioned that red blood cells must be kept within a healthy range. We don't want too many red blood cells. We don't want too little red blood cells. Why do we not want too little red blood cells? Why is that dangerous? What could that lead to clinically? Too little blood cells count. When the red blood cell count drop. What do we call that? When red blood cell count drops? Anemia. Excellent. So why is anemia dangerous? Excellent. So when your blood is not carrying enough amount of oxygen, that is called hypoxemia, meaning that the blood has a low level of oxygen, doesn't have enough oxygen. So anemia leads to hypoxemia. Excellent. So why do we also not want too many red blood cells? Now that is hyperoxemia, or now that's gonna, when you have so many red blood cells, what could go wrong? Who can speculate? Isn't the more the better? The more the better. So if you have, this certain amount of red blood cells, the more the better. If I double the number of my red blood cells, that should be better, shouldn't it? Yeah. So now your blood is gonna be like honey, so thick. So why is that dangerous? So we still need to answer the question, why is a blood that is too thick? We don't want a blood that is too thin, and we don't want a blood that is too thick. Why is that dangerous? Yes. Excellent. So when the blood is so thick and hypercoagulable, it's going to be moving so sluggish and so slow. And oxygen is not going to be arriving in time because the blood is moving so slow because it is so thick. So the blood is not getting to the tissue. The tissue is waiting for oxygen. Oxygen is very important. The cells cannot wait. They will suffocate and they will die. Even though we have enough oxygen, but because so many red blood cells, the movement is so slow, we're not going to be able to get oxygen to the tissue. Excellent. Excellent. Very good. And the same thing, you see the red blood cell? Where did the red blood cell come from? It came from this pluripotent stem cell. You see, this pluripotent stem cell is amazing. It can become a white blood cell. It can even become a red blood cell. Based on who? What makes it differentiate into a white blood cell? What makes this particular, you have one student who's going to become an engineer, another student, a medical doctor, another student, police officer, but they're all human beings, but they had different potentials. Here, the same stem cell. You see, this is my favorite figure. Let's go to this one. This one. The same cell, the same human being becoming an engineer, becoming a medical doctor, a pilot, a police officer, a firefighter. What makes it go in all of these? What guides it to go in all of these directions, including even becoming a red blood cell? You see, you see the red blood cell right here. Red blood cell. We ended up with a mature red blood cell from a pluripotent stem cell. So what guided that trip? Yeah, the cytokines, excellent. So we need to have a red blood cell cytokine. Once the red blood cell cytokine binds to a receptor on the surface of the pluripotent stem cell, this is a red blood cell cytokine. There's another cytokine for a white blood cell who will terminally become a neutrophil. That's a different cytokine. Another cytokine for one that will become a eosinophil. You understand? So once they bind, the pluripotent stem cell is going to be guided to go on that fixed, irreversible, no U-turn, no going back. It's going to be terminally going to become a red blood cell. Is that clear? Okay. All right. So this iron metabolism, this is showing us how when you ingest iron, females in the reproductive age, they all the time suffer from anemia unless they take their vitamin supplements, unless they take their iron supplements, or one of the women. Why is that? Why are, I gave you a keyword, reproductive age. How could reproduction lead to a depletion in red blood cell count for young females? Yeah. Exactly. So because of the menstrual cycle, there's a lot of blood loss. It's like a hemorrhage. just like a gunshot wound or like a stabbing wound. There's a loss of blood. So we need to replenish, as women, we need to replenish the iron count. Why do we need iron? You know, I showed you the twins, two alpha, two beta globular proteins. On the center of each protein, we find a one red colored what? We find a one, you see the two sets of twins, two alpha, two beta, but nevertheless, each core has a one red what? What's this red button? That's iron. So if you don't have iron, you cannot make heme. So why they call it heme? Heme is iron. That's why they call it hemoglobin. Globin is protein, heme is iron. So it's like when you have a hybrid, two in one, heme and a globin. So we need a lot of, not too much, obviously. We don't need too little or too much of iron. We need the right amount of iron so we can replenish this red blood cell count that we lost. So, you know, there is a lot of anemia that comes with the menstruation, especially young women who don't take the iron pills. You will see their blood count is always low and they're like very pale, and nails are really white in color, and there's a depletion of oxygen all the time, headache and weakness, and that could all be avoided with just taking the iron pill. That is found here in developed countries, but in undeveloped countries in Africa and Latin America and Asia, unfortunately, it's not very accessible. So most of the young females, they're always suffering from anemia because, you know, there's a sharp drop in red blood cells. Okay, now this iron that you take from the pill, the iron, this is too specific. This is too specific, but I'm just gonna say it really quickly. It's really not important if you know this, but it's just interesting that we take it in the form of ferric. Ferric is an oxidation state +3. Your stomach is gonna activate it and convert it from ferric +3, ferric, iron, oxidation state +3 is gonna convert it into ferrous, and that is iron in oxidation state +2. That's the active form. That's the one that's the red in the red blood cell, in the globular. You know the twins with the red iron? That's the one, not this one. So, you know, that's people also who do, you know, the gastric sleeve surgery, people who suffer from obesity and they cut the stomach. So they have a big problem converting iron ferric into iron ferrous, and that's the active one. So that just shows you the life cycle of iron. We turn it into from ferric into ferrous, and then it gets stored in the liver. So this shows you how significant your liver is, because your liver is going to be storing this iron. So whenever we need to make red blood cells, we go to the warehouse in the liver and we just pick it up from there. And then we make all red blood cells. Okay, vitamin B12 is very important in the making of this red blood cell, in addition to folic acid. So there are folic acid supplements and vitamin B12 shots that, you know, patients take. Like I said, the patients who underwent a surgery where they removed part of their stomach due to obesity or due to stomach cancer sometimes. So they need to take a shot of vitamin B12, because where do we get the vitamin B12? From the stomach. The stomach is the one who makes that. So this is the erythrocyte homeostasis. This is a very, very important figure. You really need to know this one. Why? Because it's showing you that it is the bone marrow who's going to be replenishing your supply of the red blood cells. So Remember this diagram? We said there's gonna be a receptor who's gonna send an input sensory signal. Then there's gonna be a processing center. Then there's gonna be an output. Remember that. So the same thing. When there is a drop in red blood cell count, so now we are worried about hypoxemia. An input is going to be sent to a computer center, and that's your bone marrow. That's your bone. That's your bone marrow. You see the bone marrow? And the bone marrow is going to be making these red blood cells. So that's an example of what kind of feedback, positive feedback or negative feedback. Positive. Positive feedback is when you intensify, like the baby suckling the nipple for milk, more milk coming out. Or the baby pushing the cervix, the womb is and the uterus is pushing out the baby some more. That's positive feedback. But this is not negative, this is not positive feedback. Negative feedback is when you compare before and after and they're opposite. So when the baby is suckling the milk, the baby had milk coming out, and now even more milk is coming out. That's positive feedback. But here, you had low red blood cell count. You sent the signal to the headquarters, to the bone marrow. Now more red blood cells are coming out. So now they are not the same. They're opposite. So that's negative feedback. So what's interesting here is that it is your bone marrow who's responsible for making the red blood cell. Nobody knew that. People just thought that the bone marrow, your bones are just mountains of calcium salt. They are just, minerals. But when they sliced them in half, they saw that in the core, there is the center for making red blood cells. So that's what happens. Let's look at the cycle. Hypoxemia. There is a drop in oxygen level. Who's gonna sense that? We need a sensor. Who's gonna sense that there's a drop in oxygen level? The liver and the kidney. And the liver and kidney are going to secrete this very important hormone known as EPO. EPO. What is EPO? What is EPO? EPO stands for erythropoietin. Erythropoietin is a hormone that will go and bind to receptors on the surface of the bone marrow. So EPO hormone is gonna bind to EPO receptors. So EPO hormone binds to EPO receptors. When the EPO hormone binds EPO receptors in the bone marrow, what is that gonna do? You're gonna squeeze out red blood cells. Okay, so you should always remember that when there's a drop in oxygen, EPO is going to be secreted, and EPO is going to stimulate the bone marrow to synthesize and make red blood cells. Is that point clear? This is one very important figure. You're going to see this all over the place. Beyond this course, you're going to be seeing this erythropoiesis homeostasis. What about smokers? You see, smokers, the problem with smoking is that it prevents hemoglobin from unloading oxygen. So imagine you are in an Uber ride and you want to come to school. But once you get out to the parking lot, your Uber car is cornered, sandwiched between two cars. So you cannot open any door to get out. So you are outside the parking lot. You cannot open the door and come out. That's exactly what nicotine does to the red blood cell. So nicotine prevents the red blood cell from unloading oxygen to tissue. So what's going to happen to the oxygen level in blood? It's going to go down. What do we call that? Hypoxemia. The bone marrow has no way of knowing that. The bone marrow has no way of knowing that, hey, we do have red blood cells. They just cannot get out because of nicotine. So what is the bone marrow gonna do when it senses there is no oxygen in blood? It's gonna make more red blood cells and more red blood cells and more red blood cells. But we do have red blood cells. They just cannot unload the oxygen. Exactly. So that's why when you take a blood sample of a smoker, what are you gonna find? It's really, really hypercoagulable and thick. And that's why they have all the time heart attacks and they have all the time blood vessel occlusion. And this is why, in the hospital, many of the young pediatrics, 17 years old, 16 years old, 15 years old, coming to do blood work, they have a hypercoagulable blood. Why is that? Because they're smoking in the toilet. And the mom would say, oh, not my son, not my daughter. Oh, no, they wouldn't do that. But it is because of juvenile smoking cigarette. And that's going to cause the nicotine to prevent oxygen from unloading to the tissue. And then the bone marrow is going to say, there is no oxygen in blood. Let me make more red blood cells. There is oxygen. There is red blood cells. We don't need any more red blood cells. But it would never know. There's no way of knowing that. That point there. You should be able to tell why is a smoker blood hyper thick and hypercoagulable because of the nicotine preventing oxygen to be unloaded and the compensation. Who's compensating? The bone marrow is going to be compensating so many red blood cells to make up for this loss. Okay, so now this is another amazing figure. So that shows you in your small intestine, amino acids, vitamin B12, folic acid, they help with reabsorption and the making of red blood cells. And then red blood cells are actually made in the bone marrow, in your pelvis, in your humerus, these very big bones. And then the erythrocytes are going to circulate for 120 days. How many months is that? 120 days. That's 4 months. So the life cycle of a red blood cell is 4 months. That's why a patient with diabetes is got to do blood work every how often? Every 4 months. So why is that? Because the diabetic patient, they have so much sugar in their blood that it's gonna start to latch on the surface of the red blood cell. Sugar. So much sugar. So that is called HB. What does HB stand for? Hemoglobin. HbA1c. What is HbA1c? HbA1c is what's the percentage of red blood cells in your body that have sugar on them. So the healthy range is up to 5.6%. If you have up to 5.6% of your red blood cells have sugar on them, you don't have, you're healthy. But if you go from 5.7 up to 6.4, that's known as pre-diabetes. You are on the way. You are on track. You are on a slippery slide to having diabetes. That means from 5.7% up to 6.4%.